In this video, we are going to review cell biology, and the important topics are listed here for this topic. We have microscopy, the idea of the light microscope as opposed to the electron microscope, like the benefits and drawbacks of each. And then the process of cell fractionation is an important concept for this chapter, and looking at why a scientist would undergo this process or carry out this process in the study of cells and cell parts. And then the idea of prokaryotic versus eukaryotic cells, major differences. Also animal as opposed to plant cells, again looking at major differences between those two cell types. And then another concept is the endomembrane system, looking at all of the parts that are involved in that system and what kind of proteins are produced in that system as opposed to proteins that are produced by other means. Okay, so we'll take a look at the difference there, and then you just need a basic understanding of the function of each of the organelles. We are going to be looking more in depth at the organelles, particular organelles, as we move through the chapters. So for this chapter, you really just want to have a basic idea of the major function of the organelles that I have listed. Okay, so cell fractionation, we'll go to that first. And if a scientist would want to study or would like to study a particular cell part, the best way to go about doing that is to, of course, isolate the tissue from the organism that the particular scientist would like to research, and then homogenate it, which would mean really, breaking those cells, that tissue apart, and opening up all of the cells to release the cell contents. And then you go through this series of centrifugations whereby different parts are extracted or can be extracted based on weight and size. And I'm not going to have you memorize the recipe here. You can see, for instance, to obtain ribosomes, you'd go through 1, 2, 3, 4 different centrifugations and different times and different speeds to obtain those ribosomes. This is something you can look up. It's a recipe kind of idea, but just the overall idea here is cell fractionation is a way to, you can say, fractionate or break apart cells and isolate a particular cell part so that you can study it further. And that's the why. You want to study a particular cell part and research that particular cell part. And then the idea of microscopy. So a light microscope is wonderful in that you can view living organisms. So for instance, here we have a euglena, a single-celled organism, lives in fresh or aquatic environments and freshwater. And you can view organisms under the light microscope up to 1,000 times their original size, and that's what's shown here for the euglena. Now, the electron microscope, you can view a lot more detail. You can get a lot better visualization of the organism. And here, this is the euglena again. And you have the scanning, which is really just looking at the outer surfaces of the organism, and then you have transmission, where you take thin slices of the organism and you look inside. But the drawback here with electron microscopy is that you can't view living organisms, only organisms that have been preserved. Because you are using electrons in a vacuum, which is the electron microscope mechanism to view these organisms. And of course, in light, you're just using wavelengths of light to view the organisms. So electron microscopy, good because you can get a much greater magnification, much greater resolution, but you have to kill the organism in the process. So benefits and drawbacks of both, depending on what research you're wanting to carry out. Now, prokaryote versus eukaryote, we have an example here of the major size difference, so you can just see that right off the bat. The prokaryotic cell, much smaller than a typical eukaryotic cell. Now, you are made of eukaryotic cells. Your body is made of eukaryotic cells. But you also have prokaryotic cells in your body, mostly in your intestines or in your intestinal tract, and then also lining the surfaces of your body. And you actually have 10 to the 10 times more prokaryotic cells in and on your body than you do have eukaryotic cells. That is pretty amazing. And the big reason for that would be, or the possibility of that, is that prokaryotic cells are a lot, very, very much so smaller than eukaryotic cells. And a lot simpler. They don't have as many organelles, as you can see from this particular slide, whereas eukaryotic cells have a lot more complex, compartmentalized structures called organelles. They actually have a membrane around the genetic material, but both prokaryotic and eukaryotic cells have some kind of genetic material giving them the instructions, and they also both have proteins, which are either structural or enzymatic function in the cells. Okay. And then we'll skip on to the endomembrane system. Now, with the endomembrane system, you're making proteins that are going to leave the cell. A great example is insulin. Insulin is a protein that's also a hormone, and it's made by cells in your pancreas. And it's made through this endomembrane system because it is secreted to the exterior, and it's used in your bloodstream in the regulation of your blood sugar. So it's made on the ribosome, on a bound ribosome, a ribosome that is bound to the rough ER, and then through a transport vesicle, it moves, here's our transport vesicle, to the Golgi apparatus, where it is processed and finished, and certain side groups are added and such. We're not going to get into the specifics of those modifications, but just to know they do occur in the Golgi. And then another transport vesicle buds from the Golgi and then fuses with the plasma membrane, releasing those proteins to the exterior. Now, if the protein was going to stay inside of the cell, we would have the protein processed on a free ribosome, so a ribosome in the cytoplasm somewhere out here. And then it would stay inside the cell. It wouldn't go through this transport process, this endomembrane system. So I want to show you or animate this endomembrane system process. And this is another animation on Isidore. So let's go to endomembrane system here in the week 2, chapter 6 folder. And let me just orient you to this, because no, these are not hot dogs or sausages. This is a cell, and here we have our nucleus with the DNA, and here is the rough ER with the ribosomes, and here's the Golgi, and here's the cell membrane or the plasma membrane. Okay, so we're going to start in the nucleus. We're going to make RNA copies of the DNA, and the RNA is going to travel to the ribosomes, where we're making our proteins that are going to be exported. Here comes the transport vesicle. Process those proteins, add side groups, and as you can see, this protein is released. Another type of protein that's made through the endomembrane system is a structural protein that could stay in the plasma membrane and serve a function there. And then a very special case for the endomembrane system is the production of an organelle called a lysosome. So these would be digestive enzymes produced. And this would actually, and again it's like an exception for the endomembrane system, would actually stay in the cell and function in the cell. So that, I believe, is the end of our discussion here for cell biology. If you have any questions, please contact me.